This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 213, recorded on March 12th, 2020. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. Hello, everybody.
Good to hear you. It's been a while. I'm sorry.
It has. I miss you guys.
But now we're back on our schedule. Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
And we're both here.
Yes.
We're healthy.
Yes.
And
so far,
ready to talk about microbiology. And we're going to talk about SARS-CoV-2, the virus, but we're also going to do something different, a bacterial story. So take your mind off of SARS-CoV-2.
It's a good idea. Good idea.
You know, can be thinking about it too much. So if you like what we do here on TWiM, consider supporting us financially. Go to microbe.tv/contribute. More on that later. All right, let's start with a copper story from Michael.
Well, this is a snippet, and I think everybody has been asking me lots of questions about the effectiveness of hand hygiene. How do we need to clean our homes, surfaces, et cetera? So I thought this was right up our alley. So as we all know, we're in the midst of this global pandemic caused by an emerging pathogen, SARS-CoV-2, that is literally paralyzing our globe. And it's this uncertainty that I'm hoping that this paper will, assuage some of your fears. And we have heard countless times for the need to distance ourselves, cover our cough, control our sneezing via tissues, and our poor elbows and crux of the arms we're continuously coughing into. But we all know that there are these things out there called fugitive emissions, things that we can't control, that escape despite every effort that we make. And so one question that I've been getting asked quite frequently is, do I suppose that the SARS-CoV-2 will be less contagious in warmer weather? Well, simply put, I always answer it's the physics of how droplet secretions, which contain this particular virus, remain aloft. And it has less to do with the temperature sensitivity of the virus, but more so to do with the humidity that is in our air. In general, in the winter months, when the temperatures are colder, we turn on the heat, which dries out the air in our homes and offices. And we have all been shocked by, at least I have, by my little brothers when they discovered that aspect of static electricity in physics. Then, if you extrapolate, when someone coughs or sneezes into the air in an uncontrolled manner, rather than their sleeve, these aerosol droplets are sprayed and kinetically pushed into the air. And in the winter months, these things can remain aloft because the air holds less moisture, and those little particulates can remain floating around for long periods of time. And then depending upon where they land, the virus may be stable. And in the case of the corona family of viruses, they're stable on inanimate surfaces for between 72 and 96 hours. And Vincent pointed out that paper in one of his tweets a few weeks ago. And these were data that were done long ago in 2018 about the stability of viruses and bacteria on various surfaces. Now, but in warmer months, when the air is more humid, these small droplets then fuse with the droplets of humidity. They become larger, and then gravity pulls them to the ground faster. So you don't want to let your little toddlers crawl around on the ground because they may be picking up these viruses. And the more humid the air, the faster the droplets fuse. And this is why, as Amy Chang-Volmer posted in her feed, and Amy is at Swarthmore, she posted, and I'm quoting, This is why a humidified bedroom in the wintertime, even when the heat is on, is a method we have to limit respiratory infection. So today's snippet is principally going to talk about how things like the SARS-CoV-2 virus is spread, and a solution that we might already have in our infection control's bag of tricks to control their spread. And the paper is in an open access journal. It was published in 2015, and it's entitled "The Human Coronavirus 229E Remains Infectious on Common Touch Surface Materials." And this paper comes from the University of Southampton in England, and it's by Warnes, Little, and Keevil. But while it's 5 years old, its findings are especially relevant here today. So we're going to go straight to the figures. We're not going to review how bad SARS-CoV-2 is out there. We know we have upwards of almost 130,000 cases at this point in time. But in the first paper of the figure, we find the persistence of infectious human coronavirus on common surface materials.
And let me point out that this is a strain 229E, which typically causes common colds. So that's why they're working with this one, so it doesn't have to be high containment facility.
Correct. And more importantly, they're actually measuring infectious particles. This is
Thank goodness.
This is in stark contrast to much of the literature that has come out in the last 4 weeks, where they're just measuring whether or not RNA is present. So you're looking at infectious particles. Now, they start out with 1,000 plaque-forming units, so that's likely what we're going to encounter on a surface from someone who is coughing or sneezing, and they're actively shedding virus. And they looked at common things that we have around our houses, homes, and more importantly, in our hospitals. So it's common plastics like PVC, ceramic, your Corian countertop, glass like your cell phone, silicone rubber like your cell phone is encased, rubber and stainless steel. So common household items that each of us come in contact with on a routine basis. And in stark contrast, we see that the corona family of viruses can remain viable on surfaces up to about 5 days. And we think the infectious dose for the SARS-CoV-2 virus is approximately 10 to 100, Vincent. Have you read anything to the contrary?
Actually, I haven't read that at all. I think it's probably much higher.
It's much higher. Okay.
I would say it's at least 1,000, if not more. Right.
So that's even better, because we know that it will then, as we get into the meat of the paper, it will then begin to show how effective some of the solutions that we can already put into place will become.
I think that's an important point, is that while this has some infectivity after 5 days, it's probably too low for transmission. And my feeling would be if 1,000 is enough, and I think for influenza, in some cases, you need 10,000 infectious particles. Probably by 2 days, it's really low enough so it won't transmit. And that's a caveat with all these studies, is that even though you could detect 10 PFU after 5 days, that probably would not infect you.
Yeah. So the bulk of the paper is encased in the next series of experiments, where they simply ask the question whether a brass or copper nickel surface at room temperature. So it's, again, important that they're looking at plaque-forming units. They're measuring viral particles that can inactivate cells. And they are asking the question, will copper inactivate? So they take their 1,000 plaque-forming units, and they have a simulated wet droplet. So it's effectively, if you will, a cough or a sneeze. And it's deposited on the coupon. And then they ask the question, how quickly do the viruses become inactive, that is not able to form a plaque? And what they found is that the coppers and brasses, the higher the concentration of copper in those alloys, the faster that the virus was inactivated. And since this is open access, you can go and peruse the data at your leisure and actually ask yourself the question, is a copper surface better than a stainless steel surface? And one of the things that you immediately notice in their figures is stainless steel looks like a flat line. The virus just sits there and says to the stainless steel, impress me. Nothing's happening. Whereas on the copper surfaces, the higher the concentration of copper, the faster it inactivates. And then the next clever experiment they did, they used the same sized inoculum, 1,000 PFUs, and they applied it in a much more focused area, 1 microliter per centimeter squared, and then they dried it. So this is effectively simulating how a healthcare worker may pick it up in a hospital if they haven't cleaned the room properly. And what they learned is that the copper was even better at inactivating fingertip type fugitive emissions, where you'd likely pick up the virus with your hands and then you would interact with your mucous membranes, your eyes or your face or someone. And they have done these experiments multiple times, illustrating how well the copper how the copper is able to inactivate it. And in fact, in this finger touch experiment, the copper was approximately 8 times faster than when they looked at it when they were simulating the wet cough or the wet sneeze.
Less than 5 minutes.
Yeah.
What is cartridge brass, Michael?
Cartridge brass is an alloy of zinc and copper together. It's what bullets are. The cartridge that your bullet is in.
It's pretty good also for inactivation here.
Yeah.
Actually, it's more effective than just copper, isn't it?
Yes. The zinc is thought to, because zinc is also another transition element. And when you make an alloy, you're effectively creating, if you will, a new metal for the transition electrons to facilitate the generation of the free radicals. And Warren's, Little, and Keevil go into great detail explaining the mechanism of how this works. But the short answer is the copper liberates free radicals, whether through a Fenton-like reaction, where the copper 1 and the copper 2 interact with oxygen, and they make hydrogen peroxide, singlet oxygen, hydroxy ions, and they then serve to inactivate the virus through destabilization.
Doesn't it take water? Doesn't it take water to carry out those reactions?
There's enough moisture.
It happens in dry conditions.
That's effectively the almost the contradictory, counterintuitive aspect. When it was dried, the copper is still able to affect this killing mechanism. And they actually proved that the copper is actually generating free radicals, even in spite of Elio's disbelief that you need a bucket of water, because they added.
I didn't say that.
Well, almost.
Just any water.
Well, they have a great control in their experiment. And, in our rush to get a lot of the data out into the literature about SARS-CoV-2, many folks are short-circuiting their controls. But here, because this science was done 5 years ago when the reviewers were looking for better controls, they add 2 chelators that can actually go after and mop up the copper 1 and copper 2. And the 2 chelators that they use, the first one is good old EDTA, ethylene diamine tetraacetic acid, that sups up copper 1, and then bathocuprone disulfonate, BCS, that mops up copper 2. And they were able to show that if you add those 2 chelators, you can actually shield the virus, but eventually the copper overcomes the chelators because it likely consumes the chelator, and the copper then inactivates the virus. But one of the really interesting pieces of data that they snuck into this paper is to show how it changes the structure of the virus. And they give us an EM snapshot showing us that a virus on stainless steel looks like the garden variety coronavirus. And then when it's exposed to copper, it becomes fundamentally different. And if we know anything about viruses from listening to TWiM, we know that viruses have to have the right shape to be taken up by the receptor in order to get into the cell. So even if the virus is still able to plaque, you may be effectively increasing the concentration of virus that you need in order to slip a good one in, because exposure to copper will likely change its spots.
Really trashes them.
Yeah, it really does.
Amazing.
And there was a preprint that was done by a group out of the National Institutes of Health that it's on the bioreprint server.
It's on medRxiv now, right?
Yeah, it's medRxiv.
So there are two of them now. There's bioRxiv and now there's medRxiv.
Yes.
Another place for us to check.
Another place to drive us to drink. And this particular study has actually looked at the current SARS-CoV-2 virus, and they were able to show that copper is able to similarly do it. And the last piece of Bill's experiments that I want to talk about is showing that the copper exposure to the copper surfaces actually results in the degradation of the viral nucleic acid. And that would be good news because it would reduce the false positives that many of the PCR assays may be detecting. And because for the most part, our screening system is using PCR to assess whether or not patients have been exposed. And finally, as this is a snippet, I want to tie it into a nice little package for you all. There was a paper that came out in JAMA as a research letter showing you the hotspots.
JAMA is the Journal of the American Medical Association, right?
Correct. I apologize for that. I should have given the acronym. I can beat myself now for not explaining acronyms. And the research letter came out of a lab in China where they literally went around a hospital room and they asked the question whether or not the SARS-CoV-2 virus was in air, and they were sampling the air. They were looking at surfaces and personal protective equipment. And again, you can download this paper. I believe it's in open access, as most of the SARS-CoV-2 viruses, but they show an interesting hotspot map of where the virus is in the hospital room. And what we find is there's quite a few of these viruses on the floor. So again, if you have to tie your shoe, make sure you wash your hands subsequent to playing with your shoes, putting them on, taking them off, and before you interact with your face, because the floor seems to be a hotspot, as is the toilet, which also implies that there is some shedding of the virus in fecal. So it could be fecal oral. So another reason to wash your hands. So I think I'm going to conclude the snippet there, Vincent, unless you have any questions.
Well, I have a question. How close is coronavirus from the paper to the one we're talking about?
Well, in the
can you extrapolate easily, or is it a stretch?
You probably can extrapolate it easily that the copper will inactivate it, because copper can inactivate norovirus, which is refractory to everything but
totally
bleach.
Unrelated. Sure. So what's your suggestion? That people just use copper surfaces for everything and remove what surface they have?
I hadn't intended to talk about this, but I published a paper in Applied Environmental in January that shows that industry has been able to apply copper to common bed surfaces. So if I was standing up a SARS-CoV-2 hospital and you had a large number of infected patients, I would look at using a copper surface bed to limit the fugitive emissions that a healthcare worker might encounter. Because our biggest risk, and we all have seen the New York Times /CDC curve, where we're trying to suppress the peak so that we can preserve our healthcare workforce. Because right now, our only solution, if someone's been exposed, we put them in self-isolation for 2 weeks until we get sufficient testing up and running. And then if they do convert, it's another 2 weeks until they have 2 negative PCRs that shows they're viral free before they can return to work.
I didn't realize that. It takes a whole month. Huh?
Yeah. If they're exposed and they do contract the virus, irrespective of how sick they become, because we don't want those individuals shedding virus to be interacting with those who are vulnerable and ill.
So Michael mentioned this preprint, which is at medRxiv, and it should come out in the New England Journal of Medicine any day now. But they also show that SARS-CoV-2 and 1 are both inactivated on copper surfaces. So, the half-life on copper is 3 hours.
Oh, my.
So, half-life is important because that means
But that's RNA, though, Vincent.
This is infectious virus, no?
I don't know about the medRxiv. I haven't dug into the methods on it yet.
Let me look here. I thought it was infectious virus, but I want to look because we don't want to mislead people. Methods. They did TCID50s. They're doing infectivity assays.
Okay.
So that's good. 3 hours on copper. So that means in 3 hours, half of the infectivity is lost. It depends how much is there to begin with, but you can get an idea that in a couple of hours you start to lose it. But what this tells you, and cardboard is 8 hours, steel 13 hours, plastic 16 hours. So the virus can last a good amount of time on surfaces. And what that tells us is that, so if it were 5 minutes, then we wouldn't worry. But now it means you can contaminate your hands. You should wash your hands all the time, basically.
But they are using, in fairness, 4 orders of magnitude greater amounts of virus. They're using upwards of 10 million infectious particles.
Yeah, which is good because they're calculating a half-life, right?
Yes.
So the half-life should be independent of how much virus you put on, right?
Correct.
So this tells you that if there's a contaminated surface and it's copper, in 3 hours, half of the infectivity will be lost. But you're right. If you started out on the copper with 1 million PFU, it might take quite a few half-lives for it to go away.
To be safe.
To be safe. The only good outcome of this would be if it were instantaneously, 5 minutes gone, and it's not. So this tells you surfaces can be contaminated. So therefore, you need to wash your hands so you don't risk inoculating yourself. I mean, really, that's the bottom line. And they also looked at aerosols in this paper, artificially generated aerosols, and they can last a couple of hours too, half-life. Yeah. So this kind of what we know, it's transmitted by droplets, but there it's hard to do anything about because unless you socially distance yourself, which I understand Elio is doing, right?
Well, sort of. I live in a senior residence with 500 people, and so we're at risk for 2 reasons. There's lots of us, and we're old. But so they're cutting down on a lot of activities. There's all gatherings of large numbers of people are curtailed. Yeah. But Mike, I have a question. Short of remodeling your kitchen, is there something that's relevant to the home in these considerations?
You can channel your mother. Warm, soapy water and wiping down your countertops and common touch surfaces is probably your best bet because, after all,
but no copper.
No, you don't need it in your home because you have the ability to apply warm, soapy water to various surfaces with a little bit of elbow grease, and it's an envelope virus, so soap and water will inactivate. If you happen to have
the soap will dissolve the envelope,
Yes.
the fatty.
And if you have some of these disinfecting wipes, whether or not they be hypochlorous based or whether they're quaternary ammonia, we know those are very effective. But all of those companies can't yet make a public health claim because they haven't been evaluated using the SARS-CoV-2 virus. That's probably coming in weeks to months as they begin to test the virus in their various EPA sanctioned tests in order to get a public health claim under FIFRA, which is the Fungicide, Insecticide, and Rodenticide Act.
That's amazing. I'd never heard of that.
You never heard of, that's how
FIFRA.
You determine whether, that's why Perel got into trouble because they hadn't evaluated their hand sanitizer for the virus they were claiming it would inactivate. So the EPA said, you can't say that until we have validated it. And that's principally to prevent, harm to the public of, some of these snake oil salesmen out there selling you the magic elixir that will protect you from all things evil.
I think it's great. I think that's a function of these agencies to protect us, right?
Yes, absolutely.
And so, EPA, you should give them money, let them work. Don't cut it.
Yeah.
It's important stuff. I do think that governments are there to protect the citizens of the country. But some people feel otherwise, unfortunately. All right. Thank you, Michael.
So that's done on SARS-CoV until we get to the letters, and now we're going to have something completely different.
Michael, do you have a case in Charleston yet?
We have 6 in our hospital.
So that means there are a few hundred people out there with infections walking around.
Yes. One of our healthcare workers was on holiday in Italy, and she came back.
Wow.
She self-isolated herself, and she indeed converted. And that's how I know you need 2 negative PCRs before you can return to work.
Mm-hmm.
That's the CDC guidance on that.
Yeah. I just want to say that that's good, but sometimes you do get a false negative.
Yes.
It happens. Nothing's perfect. The paper is something completely different. So if you want to take your mind off of SARS-CoV-2, by the way, that's the name of the virus, and the disease is COVID-19, coronavirus infectious disease 2019. A lot of the news mix it up. They use COVID-19 for everything.
Yeah.
Not that it matters all that much, but I think you should be precise in your language. This is a paper published in Cell, and it's really a topic very close to Elio. He loves this. Peptidoglycan production by an insect bacterial mosaic. And this was sent in by a listener a long time ago, and I've forgotten who it is, so I'm sorry, but I hope you're listening still and you'll enjoy us talking about it. May have been Hannah. Thank you, Hannah. This comes out of University of Montana, Caltech, Rocky Mountain Labs in Hamilton, Montana, which was also part of the copper study that is in preprint by medRxiv. University of Sheffield, first author Deanna Bublitz, and the last author John McCutcheon.
From Montana.
Do you know any of these people, Elio?
Yeah, I know Victoria
Orphan
from Caltech and John McCutcheon from Montana. They're fabulous people.
I know.
They do nothing but fantastic work.
So this is actually a topic that we've talked about before. I think Elio has talked about the mealybug symbiosis, right? Before on TWiM, yeah.
I think so.
Yeah, so this is...
If not, I should have.
I'm sure you did.
No,
yeah.
I think you did.
I think you did. So this is about horizontal gene transfer, which we're very interested in, moving genes from one organism to another, not by being born, which you get your genes from your parents, but outside of that, vertical inheritance. And
May I add one thought?
Yeah, sure.
I don't know if it's generally understood that this is one of the major drivers of evolution.
Right.
This is not just an occasional little thing that happens between strange bugs. This is a widespread phenomenon. Horizontal gene transfer happens and has happened repeatedly and is just a big deal.
Sure is. So there's a beautiful example of it in the mitochondria and chloroplast. Would you call that HGT, Elio?
Sure.
Yeah. They were once bacteria, and they've lost a lot of genes, but they retained some, and some of them went into the nuclear genome as well. So clearly, and in fact,
most of them.
Clearly, this is one of the things they're interested in, these individuals, of trying to see, can we see the beginnings of the making of an organelle in these complex symbiotic relationships between bacteria and eukaryotes. So they work on the sap-feeding insect and the endosymbiotic bacteria within them. And as I said, we have studied these before. They're quite interesting. The association is long-lived. The endosymbionts have probably been in there for tens or hundreds or millions of years, and they're transmitted from one generation to the next, and they have undergone extreme gene loss and genome reduction. So these bacterial genomes are quite small after all these years of living with their host. So the organism, if you will, here is a mealybug, Planococcus citri mealybugs, and they have within them endosymbionts. They have a gamma proteobacterium called Candidatus moranella endobia, or moranella, and that gamma proteobacterium resides in the cytoplasm of a beta proteobacterium, Candidatus tremblaya princeps, or just tremblaya.
Isn't it wild? A bug within a bug within a bug.
A bug within a bug within a bug, exactly.
It's only appropriate that a bug that rolls into what looks like a pill would do that. Isn't a mealybug
right.
roll into something that looks like a pill?
Yeah.
They do. Yeah. They do. Planococcus citri, mealybugs, and they drink sap, of course. And so these bacteria, by the way, one inside the other, are in the cells of the insect called bacteriocytes. And the genome of Tremblaya is very small. It encodes 120 proteins. The Moranella genome is a bit bigger, but it only encodes 400 proteins as well. So they have lost a lot of genes in the process of getting comfortable with the mealybug. And in this paper, what they do is study the biochemical pathway for the production of peptidoglycan, right? This very important feature of bacteria, important for cell division, shape, integrity of the cell. Eukaryotes don't produce peptidoglycan. In fact, when eukaryotes see peptidoglycan, they usually react by making cytokines. The innate immune response is often triggered by peptidoglycan. And, of course, the peptidoglycan biosynthetic pathway is a target of many antimicrobials. And to make peptidoglycan, you have to have a lot of genes and their gene products involved in that. And so it's been known for some time now that this mealybug genome has a number of genes acquired by horizontal gene transfer involved in the synthesis of peptidoglycan. So why would an insect like this have peptidoglycan? Well, most likely for its endosymbiotic guests, I suppose. So the paper is all about trying to dissect this pathway and see where the peptidoglycan is made and where does it go, importantly. So, yes.
May I say something you're going to say at the
Always.
end anyhow?
Always.
Let's say it twice. This paper is interesting because it puts endosymbiosis on the same level as organelle.
Right.
Yeah, absolutely.
We'll talk about this again, but let me just point it out now.
That's absolutely right.
And since you all can see likely the graphical abstract, I don't know if this cell paper is in open access or not, but generally you can see the graphical abstract that really helps you to get a sense of how Morganella is doing this magic.
Moranella.
Moranella.
Let me just summarize the graphical abstract. So you have this mealybug, right? Previous bacterial infections have delivered peptidoglycan-related genes, which end up in the genome of the mealybug, right?
Mm-hmm.
And then at some point, we have the introduction of two bacteria, the Morinella, which lives inside another endosymbiont, Tremblia, and they encode the rest of the peptidoglycan synthesis pathway. So between the mealybug and the endosymbionts, you have all the genes you need to make peptidoglycan. And the peptidoglycan, and as you will see, is going to end up surrounding the Morinella within the Tremblia. And so it's an interesting problem of how it gets there even, right? Because it's part of the genes are in the nuclear genome of the insect. So it's an interesting problem.
Not only that, but the question that arises is why?
Oh, that
Why
one
did
too,
they bother
yeah.
to do that?
Yes, that's right, which we don't learn the answer to, but
No, we don't even answer that.
it's an important
We can
question.
speculate, but we
yes.
don't know the answer.
So to make peptidoglycan in gram-negative bacteria, you have three classes of reactions based on where the enzymes are located. The first is the cytoplasmic synthesis of the precursor of peptidoglycan, and I won't tell you the chemical names. The second set of reactions involve an inner membrane-associated enzymes that flip this peptidoglycan precursor into the periplasm of the bacterium. And then the third set involves cross-linking of precursors into the growing peptidoglycan matrix, modifications, and so forth. And so three sets of reactions.
Did you just say this? But figure one in the paper is just absolutely beautiful.
Yeah, they have a very nice figure here, but most of you probably won't be able to see it. But
This is the equivalent of installing a screen door in a submarine. Because if you think about it, you have to not violate the hydrogen tight inner membrane, because that's how the Morinella is making its energy. And this flipping of these precursor molecules across the membrane using a grease equivalent facilitates the swapping of the precursors made in the cytoplasm. So they're effectively sneaking it across the inner membrane without violating that hydrogen tight membrane. That's the most remarkable thing about this, because in order for the cell to continue to make energy, it has to have a hydrogen tight membrane so you can have a proton gradient to generate energy for the cell.
That's right.
That's true for all gram-negatives, not just for these guys.
Yes.
So, Michael, we had an episode of TWiM called Screen Door in a Submarine,
Yes.
because you used that phrase many years ago.
Yeah.
I love it. So they had previously studied the Morinella genes that are involved in these pathways. They're mainly involved in the first two sets of reactions. The Morinella genome encodes some of the enzymes you need to flip the peptidoglycan into the periplasm, as I said, and also the first set of cytoplasmic reactions. But they didn't fill in all the gaps. And what's really important here also is that there are no genes for peptidoglycan biosynthesis in the genome of Tremblia. And so they say probably it doesn't have a peptidoglycan, although I would say, why couldn't it acquire it from the synthesis, right?
True.
Point.
Just the fact that it doesn't have the genes. I mean, that's what a symbiosis is all about, right? You can get stuff even if you can't make it.
You borrow the cup of sugar from the neighbor.
That's right. So the first thing they did was to look for peptidoglycan in insects. They took 1,500 mealybugs and they extracted protein, and they did mass spec, essentially, to look for peptidoglycan, and they found it. So they say, all right, these insects, in fact, do have peptidoglycan. You don't know where it is, of course, because they just take the mealybugs and grind them up and put them in mass spec. But they certainly do have the right components of peptidoglycan. Okay, so now where is it? And now this is a very cute set of experiments. What they do, a major component of peptidoglycan is D-alanine, right? In fact, it's a molecule specific for peptidoglycan.
Notice it's a D
D.
Amino acid.
Right.
And not the L form, which is found in all the proteins.
That's what most of us have, right? Mostly L.
Yes, it's all L.
And so this D-alanine is in the peptidoglycan. So they got some N15 labeled D-alanine, all right, which they bought. And they soak sprouting potatoes with the N15 labeled D-alanine. And they let the mealybugs feed on that. So they're eating the isotopically labeled D-alanine.
Heavy isotope.
A heavy isotope, right?
It's much easier than a pipette mitt. Just let the insect eat that radioisotope for you.
Right. And then after they let them feed for a week, they take out the tissue and thin section it and look. And they do in situ hybridization, fluorescence in situ. And they can see where the signal is in the cells, which is being made by the N15. And then, so they see it near or at the periphery of all the Moronella cells. And if they use N15 labeled L-alanine, you see it throughout the tissue, right? Because it's being incorporated into all the proteins, right?
Yep.
So very nice. They localize it in the periphery. So let's see. So far, we have this localization of the D-alanine at the periphery of Moronella, but not Tremblaya and not in the insects. So it's going to Moronella specifically. Okay, that's good. That's important. All right, next, they want more evidence for this, that the presence of there. The microscopy isn't so accurate. So they say they're going to use an antibiotic, cefsulodin.
I never heard of that.
It's probably a beta-lactamase.
Yeah.
Beta-lactam antibiotic. It inhibits the cross-linking of peptidoglycan.
Right. Mm-hmm. It says it targets the periplasmic localized penicillin-binding protein PBP1B. And that is a glycosyl transferase and transpeptidase that joins precursors to the growing peptidoglycan matrix in the periplasm. Does that sound right, Michael?
Yep. It's not the cross-linking. It's actually similar in mechanism to vancomycin, preventing the disaccharide from being added to the extending peptidoglycan chain.
So they're going to treat their mealybugs with that, and they're going to treat them with hypotonic media, because they know if you treat bacterial cells with this antibiotic in the presence of hypotonic media, you get membrane blebbing and cell lysis. So they treat their mealybugs with this antibiotic, and they look at it. Actually, they use electron microscopy, and you can see that the periplasm of Moronella is enlarged in the presence of this antibiotic.
Whereas that of Tremblaya is not.
That's right. Tremblaya is not. And they measure the membranes. Tremblaya has 3 membranes. They measure them. They're not affected. So more evidence that there's no peptidoglycan there. Okay. Now, what's next? So most of the genes for synthesizing the cytoplasmic part of peptidoglycan, they're in the insect genome. They're acquired by horizontal gene transfer. And so now they want to know, how does this all work? Where's the protein made? Is the protein transferred? So, one idea is that you could make the precursors in the insect cytoplasm and then transport them to Moronella, but they think maybe they're translated in the insect, and then the gene products are transported into Moronella, and there they produce the precursor, right? So you make the precursor in Moronella rather than in the insect, and then shipping it over there. And by the way, in order to get to Moronella, you have to cross 5 lipid bilayers.
No less.
3 in Tremblaya and 2 in Moronella. So that's amazing. All right, so the 2 different scenarios for peptidoglycan production, precursor production. They have an antibody to a protein that would be encoded by one of these mealybug genes that they think got there by horizontal gene transfer from bacteria. So they have an antibody to the gene product, and then that gene product is a protein called MUR-F. It's a ligase that helps to join parts of the peptidoglycan, and that functions in the cytoplasm. It's part of the cytoplasmic pathway. So they have an antibody to that, and they stain these paraffin-embedded insect tissues with it, and they find that this protein, MURF, is uniformly located throughout the cytoplasm of Morinella and in parts of insect tissue, but it's completely missing from Tremblia.
Beautiful figure 5.
It's very pretty. Very nice. So this is suggesting that the proteins are made in the mealybug, and then they're transported across Tremblia and into Morinella. And they bypass Tremblia completely, right? They don't stay there because the antibody staining doesn't find any of them there.
No, there's no track whatsoever.
It's amazing, right? So the conclusion is that you have a peptidoglycan that's built in Morinella in the cytoplasm to begin with, and it's located in the periplasm. So everything you need to make it, of course, Morinella has some of the genes, but the insect also does. Those proteins are made in the insect cells and then transported into Morinella through Tremblia. It's really very complicated, amazingly, but that's how nature is. It evolves very complex associations.
And we certainly, to come back to the why question, it's obvious. It's not at all obvious why this happens this way. I mean, why bother? Why did Morinella keep those genes? It could have done it very nicely, but maybe this is the question that people who worry about organelle formation worry about. Why is the transfer from the organelle of genes from the organelle to the nucleus?
Yeah.
It's a general question. It's not obvious.
It's likely got to do with the energy budget, and the host, the mealybug, made a decision to a trade-off to give it the precursors so that the Morganella could remain, and then its energy budget would be subsequently less because it didn't have to effectively generate the precursor molecules. And it's either managing precursors more efficiently for the host or managing the energy budgets collectively of both cells, of looking at how energy gets in and out. The way I looked at it, it's the equivalent of building a rescue sub inside a sub. So you have the sailors providing the materials to the workers inside the rescue sub, and they're building a second rescue sub because ultimately the Morinella will divide, right? I mean, their goal is to make more of these cells. But the timing, then we get into the timing, which is likely their next question, is how does the cell decide when to divide again?
Yeah. All right. That's it. Anything else, gentlemen?
It was a fascinating paper.
Very nice.
Lovely
the
paper.
Figures
Lovely
are gorgeous.
Paper. Great group of people.
Good stuff. Let me see if I can find who sent it. I'll just search very briefly here to see, because it would be nice to thank the people who sent it.
The lead contact is John McCutcheon.
Yes.
Insect bacteria mosaic. Here we go. Oh, this was sent by Mehmet, who works at New England Biolabs.
Oh, I see.
I thought this paper might interest the lovely people of TWiM. Take a look. Thank you very much. It was really good, Mehmet. Very good. And thank you for calling us lovely. That's very nice.
Well, that only applies to Michelle.
Not me, that's for sure.
Yeah.
Well, you could be lovely if you want to, but I'm not. Okay,
Yes.
Let's take some email. Let's see. Oh, very interesting. Here we have an email from Kay Hoffman at the Institute for Genetics, University of Cologne. Anyone know her?
No.
Dear TWiV and TWiM team, I listened to both shows since episode 1. Wow. You can safely assume I like what you do very much. Thank you very much. I noticed on several occasions, including last week's TWiM, that you think the word prokaryote has to be avoided at any cost. I must say,
that's Mark
not by
Martin.
me.
That's Mark Martin, we were channeling, I think.
Yeah, Elio, you don't mind prokaryote, right?
I like it very much. It's not a taxonomic term.
That's right.
It's a
not
common,
taxonomic.
ordinary language component.
I must say that I totally disagree on this, although I usually agree happily to most of your other scientific pedantries. In my humble opinion, the term should be avoided is prokaryota, together with everything else that would suggest that prokaryotes constitute a monophyletic taxon, which they clearly don't.
Amen.
However, the term prokaryote itself is not considered a taxon and is extremely useful. Many aspects of cellular life are different in eukaryotes and non-eukaryotes, and prokaryote is as a synonym of non-eukaryote is less unwieldy and should be given preference.
That's right. The alternative is 3 words instead of 1: bacteria and archaea.
Yeah,
it's what prokaryote. It's that's what prokaryote means. Period.
Without nucleus.
Yeah, but that's not in the common parlance. When we say, if you look at a textbook and you look at how often the word prokaryote appears, it appears a textbook of microbiology, of course. It appears a lot. It's just simply simpler to use 1 word instead of 3. That's all.
Mm-hmm. All right. So she continues, the avoidance of the word prokaryote makes even less sense in a context where people use expressions like arboviruses and dependiviruses, the 2 other terms that are widely considered acceptable, although these groups are polyphyletic and there are no taxa called arboviridae or dependivirales or similar. That's correct. Not every biological expression that summarizes a group of organisms has to be a monophyletic clade. Think about apes, paraphyletic, insectivores, polyphyletic, zooplankton, polyphyletic, and many other terms that are useful to describe groups of species by a common property without relying on taxonomy. Prokaryotes is just one of these terms, should not be banned if we make clear that it is not backed by taxonomy. Okay.
I love this. This is exactly what I've been saying, and it's only said much better than I can.
I agree.
Copy and paste. Copy and paste.
So I objected to it because the writer misused it in some way. I can't remember why, but
that was in January. So
yeah, it's a long time ago.
Forget.
It's a long time ago that I remember. The only thing I worry about is, so if we lump bacteria and archaea together, some people don't like that because archaea are very distant from bacteria, right? They're close.
And they're very, very different. They have different flavors of ribosomes.
Right.
On the other hand, that's if you look at the genomics. If you look at what they do and what they look like, they're awfully close.
Okay.
They share environments. They look alike. There's a lot of things that makes them lumping together. That's why they're lumped together, after all.
Okay.
Regardless of what you call them.
So if you just want to say without a nucleus, that's fine, right? That's what prokaryote means, right? Before. Right. Mm-hmm. It's very good. Thank you, Kay. The next one is from Sophia. Dear TWiM team, thank you for your input on coronaviruses, info on masks. It was our last show. Was what? December?
Yeah.
January?
It was January.
Okay.
January.
I do have some questions about what Dr. Schmidt said about surgical masks. I didn't get what he said. Could you please explain why an N95 is good to prevent TB but doesn't make a difference for viruses? Is it because if viruses are transmitted through aerosols, they will still go through the mask? I know TB microbes remain suspended in air for a while, but the N95 mask does not allow penetration. The microbes are too big to penetrate but small enough to remain suspended. Is that correct? Also, a WHO lady answering questions from the audience did agree with Dr. Schmidt's suspicion that the virus is not airborne but transmits through droplets. Great. Why is it necessary then to wear a mask while walking down the empty streets of Wuhan, or why not be allowed to walk around anyway? Also, Public Health England seems to think otherwise, listing the virus as airborne. Well, sometimes people conflate airborne and droplet, right?
Yes.
Airborne are the smaller droplets that go long distance, and the droplets that Michael mentioned fall to the ground quickly.
Yep. So there's no reason. The only time that we're recommending our healthcare providers to wear their N95 mask is literally when they're very close to a patient interaction where they're likely going to be in the immediate vicinity to effectively get a hyperdose of the virus from the infected patient, like they're doing a nasal pharyngeal swab. Then the healthcare worker needs to be in full PPE with N95 mask. I was commenting that there's no evidence that shows an N95 mask for the uninfected walking down the streets of Wuhan is any better than a surgical mask, because you're out and about in the air And the only reason to wear a mask in Wuhan is if you are potentially infected and you're concerned about infecting your fellow man. And so consequently, you want to contain it. And an N95 mask is as good at containing as a surgical mask because you're effectively capturing the droplet. The other reason the general public has to be very concerned about wearing masks is they often don't know the proper way to doff. And taking something off is the process of doffing. And that is where you can contaminate yourself.
Right.
And oftentimes what they do in a healthcare system is they have a buddy watch you. And Vincent and his colleagues who have been to the needle know that the whole megillah of putting on PPE for level 4, you always have a buddy watching you put the equipment on and taking the equipment off so that you don't contaminate yourself. And this is what is so important for healthcare workers when they're interacting with the infected individuals, that they do indeed take their PPE off properly so they don't inadvertently contaminate themselves or others. The other reason is we're short of N95 masks. There's a global shortage, and we desperately need them for healthcare workers. And it's just simple compassion that you need not run out and get yourself an N95 mask, because there's no evidence that you're going to contaminate yourself from breathing. And in fact, that research letter that we talked about in the snippet portion actually shows that even in an infected patient's room, it's unlikely that the air is carrying sufficient virus to contaminate you unless you're literally very, very close to a patient, interacting with that patient where you can get literally a face full of virus.
Of course, if you're on a cruise ship and the air is being circulated, then it's a different
all bets
story.
are off.
Different story.
Then all bets are off. Yes.
Okay, so a couple more things from Sophia. Oh, the Greek CDC advised about close contact with infected patients, defining it as spending at least 15 minutes with the infected person in less than 1 meter distance or more than 2 hours in a room closed space. I assume they get their info from WHO, but these instructions wouldn't agree with an aerosolized pathogen. Do they make sense? I really got confused. Well, I think the terminology is being mixed up because virus is definitely droplet spread, not airborne. So, but as I said, I think they're misusing aerosolized or airborne, but they should use droplet because those are the right recommendations. Speaking of WHO, they recommend hand hygiene lasting 40 to 60 seconds for hand washing, 20 to 30 seconds for alcohol rubbing. Isn't this way too long for a nurse? I mean, do nurses actually have time for this, or is this the reason they wear gloves? How often should you hand sanitize if you wear gloves? Michael, you might know that.
The answer is every time you have a patient encounter, you need to hand sanitize. And that is why the average nurse who works an 8-hour shift washes their hands for 1 hour per that 8-hour shift.
Wow. So they do have time to wash their hands, right?
It's built into their
And if
protocol.
they wear gloves, they still have
the
to
gloves
wash
are contaminated, and they're going to contaminate you.
So you take the
have to
glove
wash.
off, you throw it out, and then you wash your hands anyway. Is that correct?
Yes, because the doffing process may inadvertently contaminate your hand.
Right. Speaking of donning and doffing, my colleague Daniel Griffin made a wonderful video for healthcare workers about how to don and doff PPE. I will put a link. It's on YouTube. It's he's part of a large physician network here in the tri-state area, and they made it for them so that they know exactly how to do this. But it's good for everyone. Should you encounter a mask and you want to wear it properly, check it out. P.S. This is from Sophia. I think of Elio too when I read about mushrooms.
Hey.
There you go, Elio. A lot of people thinking about you.
Glad to hear that.
All right.
Well, we're going into prime mushroom hunting season, aren't we? As it begins to wetten up.
Well, not for most of the country. In Southern California, it is. Actually, it's almost over. But in the rest of the country, the mushroom hunting season is actually, it can start for certain mushrooms in the spring, like for morels, but the height of the season is the early fall.
Okay.
Or whenever it rains.
Whenever it rains.
All right, that is TWiM 213. You could find it over at asm.org and any podcast player. Please subscribe so you get every episode as we release them. They're all free, and we know how many people are listening. If you like what we do, consider supporting us financially. You can go over to microbe.tv/contribute, and you can sign up to give us as little as $1 a month. We'd appreciate it. Questions and comments, we'd love to get them. TWiM@microbe.tv, and we promise now we'll be back every 2 weeks for the foreseeable future. Elio Schaechter can be found at a wonderful blog called Small Things Considered. Thanks, Elio.
My pleasure. I really got a lot out of this.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.blog. I want to thank ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.